So there's two types of antigens, the A antigen and the B antigen, that can be found on the surfaces of red blood cells, aka erythrocytes. And two possible antibodies can be formed in the plasma. The anti-A antibody, meaning the antibody that binds to the A antigen, or the anti-B antibody that binds to the B antigen. So any individual with these blood types can have any individual can have various types of antigens and spontaneously preformed antibodies. And when these different antigens, agglutinogens, aka antigens, meaning they're also called agglutinogens because they can result in clotting. When these antigens are mixed with their corresponding antibodies, there can be serious hemolytic reactions. Remember what hemolytic means. Hemo meaning blood, lytic meaning lysis. So hemolytic meaning blood lysine reactions. The body, any person will reject cells, erythrocytes, or other cells, any other cells, bearing antigens that don't match their own, by producing antibodies that bind to foreign antigens or foreign glycolipids on the erythrocytes. These can promote clumping or agglutination of the foreign antigens and the cells that they're found on. So the antibodies will promote agglutination, aka clumping, of the foreign glycolipids and the cells that they're found on. So for example, a person who has an A blood type has the A antigen on their erythrocytes. Because they don't have the B antigen on their erythrocytes, they will either form or may form anti-B antibodies when exposed to the B antigen. A person with blood type B will have the B antigen on their erythrocytes and possibly have anti-A antigens, antibodies, anti-A antibodies in their plasma. An individual who has an AB blood type will not produce antibodies against A or B because their erythrocytes already have both of these antigens, and therefore both of these antigens are recognized as being part of them and not foreign. A person with an O blood type has neither the A antigen or the B antigen on their blood type, on their erythrocytes. And the result is that they will have or form both the anti-A and the anti-B antibodies. A way to determine someone's blood type is to treat their blood with either the anti-A antibody, the anti-B antibody, or the anti-D. Anti-D having to do with that rhesus vector that we'll talk about in a minute, but it's another antigen. So A blood sample, for example, is taken and it's treated with the anti-A antibody. When it's treated with the anti-A antibody, it causes clumping, meaning that the anti-A antigen is present. When their blood is treated with the anti-B antibody, there's no clumping present that occurs, meaning that there's no B antigen on their erythrocytes. However, when their blood is treated with the anti-D or the anti-RH factor antibody, they get clumping, meaning that that antigen, the RH antigen, is present. Do you think D meaning RH in this case? Another blood sample is treated with the anti-A antigen. And when it's treated with the anti-A antigen antibody, you get no clumping, meaning that the A antigen is not present in that blood sample. However, when it's treated with the anti-B antibody, we get clumping, meaning that that antigen is present. Also, when we treat that same blood sample with the anti-D or anti-RH factor antibody, the result is that you get clumping, meaning that that person has a B positive blood type, meaning their blood contains the B antigen and the RH factor. Third one down. This blood treated with the A antigen results in results in clumping when it's treated with the anti-A antibody, meaning that that antigen is present in that blood. When that blood is treated with the anti-B antibody, you get clumping again. When that blood is treated with the antibody against the RH factor or the anti-D antibody, there you get clumping, meaning that this person is AB positive. Last one here. This blood is treated with A antigen and you get no clumping, meaning that that A antigen antibody is not, the A antigen is not present in that blood. With the anti-B antibody, you get no clumping, meaning that the B antigen is not present either. When it's treated with the anti-D or the RH factor, there's no clumping, meaning that this person is O negative. Their erythrocytes do not contain the A antigen, the B antigen, or the RH factor. Remember, this D represents the RH factor. So this is an antibody directed against the RH factor that will bind to the RH factor. So what is this person's blood type? Take a sample of blood, we mix it with the 3 different types of antibodies. The anti-A antibody, when it's mixed with the anti-A antibody, you get clumping, meaning that the A antigen is present on that blood. When it's treated with the anti-B antibody, you get no clumping, meaning that the B antigen is not present on that blood. However, when you treat it with the anti-D antibody, the antibody that's directed against the Rh factor, you get clumping, meaning that this person has the A antigen on their erythrocytes and they have the Rh factor on their erythrocytes, so they are A positive. The control in this case is treating that same, I would say treating that same blood with a serum that doesn't contain any of these three antibodies. Doesn't contain the anti-A, doesn't contain the anti-B, or the anti-D. Just to clarify what clotting would look like in this blood and what non-clotting would look like in this blood. So in the control, that's a negative control, meaning you would see no clotting. So, talked about how a hemolytic reaction can cause, bear with me here, a hemolytic reaction. If you introduce a foreign blood type into a person, their antibodies against the A, B, or Rh factor will cause agglutination of that foreign blood, causing clumping of those erythrocytes, foreign erythrocytes, which can cause clots or clogs in small vessels. Also, these clumped foreign erythrocytes can rupture, and when they rupture, the hemoglobin is released, and that hemoglobin can precipitate in the kidneys, meaning it falls out of solution, precipitation in the kidneys, clogging the kidneys. Talk about that, how that would occur in the kidneys later in the semester. So let's take a minute and talk specifically about these Rh factors, or the Rh blood groups. There are about 8 different Rh agglutinogens, 3 are antigens, 3 of which are common, the C, D, and E. The presence of these Rh factors is indicated as a positive when we talk about someone's blood type. Rh antibodies are not spontaneously formed in Rh negative individuals. They have to be exposed to the Rh positive blood. Remember, our anti-Rh is the same thing as anti-D. However, if an individual with a negative blood type receives a positive blood type, they will form anti-Rh antibodies against that foreign blood. And a second exposure to that Rh positive blood can result in that typical transfusion reaction, that hemolytic and vessel clogging reaction because of the clumping or agglutination of those erythrocytes. A disease that can result from mismatching of the Rh factor is what's called the hemolytic disease of the newborn. If you think about what that that's, remember how I want you to learn these root words and base words in order to comprehend these bigger words. And so what would hemolytic disease of a newborn, what would that mean? It would mean a disease of the newborn that results from hemolysis. How do we get this hemolysis in a newborn? The mother is Rh negative. So we start with a mother who has a negative blood type. And she becomes sensitized when she's exposed to an Rh positive blood type from either a previous pregnancy with a baby who was Rh positive or a positive blood type transfusion. This will result in her body producing Rh positive antibodies. When that woman becomes pregnant again with a baby who has a potential negative blood type, actually, wait, a positive blood type. When she becomes pregnant again with a positive blood type, those antibodies that she's produced from her previous exposure, those Rh positive antibodies, can cross the placenta and attack and destroy the red blood cells of the future baby, Rh positive baby that she carries. Because antibodies, we'll talk about this in the next chapter, but IgG antibodies, like Rh positive antibodies, can cross the placenta. So the Rh positive antibodies contained within the mother cross the placenta and attack the Rh positive red blood cells of the baby that she's carrying. Let me say that one more time. So the antibodies against the Rh factor that are present in a mother with a negative blood type can cross the placenta and destroy the red blood cells of the future baby that has the positive blood type. One treatment that's been developed to prevent hemolytic disease of the newborn is, bear with me, cats, stop it, Leo, stop it, Leo, no, stop it, stop it. Stop it. I'm sorry. A drug that has been developed to combat hemolytic disease of the newborn is the RhoGAM shot, which is a drug that ties up the Rh antibodies, the anti-Rh antibodies in the mother of the baby. And so anytime that a woman becomes pregnant, doctors will give her a RhoGAM injection, meaning a drug that will tie up the Rh positive antibodies in her blood until she gives birth to that baby. And this prevents her from developing, or let's say it this way, it prevents her from passing Rh positive antibodies to the developing fetus. Every once in a while there is a baby born with a hemolytic disease of the newborn. And the ways to treat this and combat this is pre-birth transfusions or exchange transfusions after birth. Working to get rid of the Rh factor that's making the baby sick. So the RhoGAM shot ties up the Rh positive antibodies in the mother so that they can't be crossed. Those antibodies are tied up to the point of where they can't move across and attack the newborn baby, the developing baby. So the ABO blood groups are determined by the glycolipids A in the plasma membrane of the red blood cells. A person with a type A blood can safely donate red blood cells to a person with let's see here, a person with an AB or A blood type and can receive red blood cells from a person with either A or O blood type. So in this case, because we need an O, see here, can safely donate someone with a type I'm not sure I really like this question because except the most correct answer for each blank. They can't receive blood from a person who has a B type. So it can't be this one. They can receive blood from someone who either has A, AB, oh wait, no. They can donate blood to someone who has A, AB, or O. Who can they receive it from? Someone who either has B, no, not B, A or O. So they can donate to someone who has A, AB, A or AB. But they can only receive blood from someone who has O blood or A. So A or O. So our choices, we A or O. So we've got B, we've got D, and we've got E. But we know that they can't give blood to someone who has O, so that one's out. E's out. They can't give blood to someone who has B, so that one is out. So the answer has to be this one, B right here. last part of this, talking about leukocytes. Leukocytes, meaning the white blood cells, are the mobile units of the body's immune defense system. The immune system is made up of leukocytes, their derivatives, and a variety of other plasma proteins. They recognize and destroy or neutralize materials within the body that are foreign to the normal self. Anything that's foreign to what's present normally in the body. They function in defending against invading pathogens, identifying and destroying cancer cells that arise on the body. And they also function as a cleanup crew to remove worn out cells and tissue debris. Leukocytes, leuko meaning clear, are colorless because they lack hemoglobin. They can vary in structure, function, and number. They're somewhat larger than erythrocytes, and there's 5 different types of circulating leukocytes: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. All leukocytes ultimately will originate from the same undifferentiated multipotent stem cells in the red bone marrow. Granulocytes and monocytes are the only ones produced in the bone marrow. You two, stop it! I'm sorry, my cats are wanting to have a war here. Most new lymphocytes, just go on, now. So most new lymphocytes are produced by lymphocytes already in the lymphoid tissue, such as in the tonsils or lymph nodes. We will talk about that more in the next chapter. The total number of white blood cells and the percentage of each type can vary considerably to meet the changing defense needs of the body. So 5 different types of leukocytes: neutrophils, basophils, eosinophils, monocytes, and lymphocytes. So neutrophils are our phagocytic specialists. They will release a web of extracellular fibers called neutrophil extracellular traps or nets that contain bacteria-killing chemicals. So they kill by releasing this web of extracellular fibers that contain bacteria-killing chemicals. They also can kill bacteria via phagocytosis. Their other functions is that they're the first defenders on the scene of a bacterial invasion. And they're very important in the inflammatory pathway, and they work to scavenge to clean up any debris. They are the most abundant leukocytes. Eosinophils are associated with allergic conditions and parasite infections. They make up about 1 to 4% of the leukocytes found in the body. Basophils are the least numerous and the most poorly understood of the leukocytes. They're quite similar structurally and functionally to what are called mast cells. Mast cells also release inflammatory stimulating granules. So basophils synthesize, store, and release histamine. Histamine is important in allergic reactions for promoting inflammation. Heparin speeds up the removal of fat particles from the blood after a fatty meal. It can also prevent clotting of blood samples and is used extensively as an anticoagulant drug. Monocytes emerge while still immature and circulate for about a day or two before settling in various tissues in the body. They continue to mature and enlarge in resident tissues to become macrophages. Their lifespan can range from several months to years. Macrophages meaning they become professional phagocytes. Lymphocytes provide the immune defense against targets for which they're specifically programmed. This is our adaptive immune system. They live for about 100 to 300 days, and there's two types of lymphocytes. There's B lymphocytes that produce the antibodies which circulate in the blood, and these are responsible for the antibody-mediated humoral immunity. There's also T lymphocytes that do not produce antibodies, but they directly destroy specific targets by releasing chemicals that punch holes in the victim cells. So they destroy specific target cells by releasing chemicals that punch holes in those target cells. The target cells can include body cells invaded by viruses and cancer cells. Right here, this slide just gives a typical blood cell count number of normal formed elements within the blood. Remember which of the three leukocytes which three leukocytes are granulocytes, which ones are agranulocytes. Remember general ideas to what they do and what their specific functions are. How to recognize them, and their normal numbers in a normal healthy individual. So most common are neutrophils. The lymphocytes are the second most common. The monocytes are the third most common. Eosinophils are the fourth most common, and basophils are the least common. Other two formed elements, the erythrocytes and the platelets. Which of the following is not contained in the buffy coat? Well, where was the buffy coat found? The buffy coat was found in the hematocrits. The buffy coat contained platelets and white blood cells. So which is not contained there? has to be the erythrocytes, because those are the red blood cells that make up the bottom. When we make a hematocrit, they're the ones that settle to the bottom, and then the buffy coat sits on top. What are the least abundant of the formed elements? Remember the mnemonic I taught you in lab, never let monkeys eat bananas. So basophils are the least abundant. What are the most abundant agranulocytes? So the agranulocytes include the monocytes and the lymphocytes. The monocytes, no, the lymphocytes are the most abundant, because remember, they're the second after neutrophils. Never let, so lymphocytes are the most abundant agranulocytes. Agranulocytes meaning the monocytes and the lymphocytes. Which one is the most abundant? The lymphocytes out of those two. So coming back, and we've seen this graphic, we've seen this diagram a few times during this PowerPoint. Pluripotent stem cells are going to form myeloid stem cells and lymphoid stem cells. From the myeloid stem cells, we get erythrocytes being formed, platelets, all of the granulocytes, the eosinophils, basophils, and neutrophils, and also the monocytes. The lymphoid stem cells produce the lymphocytes, the T lymphocytes and the B lymphocytes, and the natural killer cells, which we'll talk about in the next chapter, in chapter 21. Okay, last slide, some true or false questions. Hemostasis is a production of formed elements in the blood. What's the production of formed elements? Hematopoiesis. So it can't be hemostasis. So that's false. Blood viscosity stems mainly from the electrolytes and monomers dissolved in the plasma. This is false because the blood viscosity stems mainly from the number of erythrocytes or red blood cells. Lymphoid hemopoiesis occurs mainly in the bone marrow. This is false. And we'll learn in the next chapter the major reason why this is false is because those cells, the lymphocytes that actively divide and the natural killers are going to do that in response to a foreign antigen. When do they see that foreign antigen? They're going to see it in the tonsils. They're going to see it in the lymph nodes. Oxygen and carbon dioxide bind to different parts of the hemoglobin. This is true. Oxygen binds to the iron. Carbon dioxide binds to the amino groups on the polypeptide chains. The liver stores excess iron as ferritin. That's true. A person develops anti-A antibodies only after he's exposed to an anti-A antigen, and anti-B antibodies only after he's exposed to the antigen B. That's false. They're preformed.